Hey guys, Nicole Mashburn here, and I'm going to talk to you today about cell division. Let me get the pen started. Okay, so for some of you who have had biology in the past, or maybe had 103 here at Calhoun, you understand a little bit about cell division. There's a lot to it. We're going to kind of do the need to know for this course. If you're a biology major and you're planning on going into biology, you need to know it in a whole lot more detail for your coursework. But for this course, this is all you need to know about cell division. So what is cell division? Why do we even want to talk about cell division? Well, the main reason is because in order for things to get bigger, to grow, cells have to divide. So think back to the very most rudimentary beginning of life where you have a sperm and an egg, and they come together. There's a sperm and egg. They come together, and you form the very first conceptus of life. So what happens is that one cell has got to divide and divide and divide till it gets bigger and bigger and bigger, and finally it's a baby. Okay? So then this baby has to divide, and the baby doesn't divide, but the stuff inside the baby is going to keep dividing and dividing and dividing until finally it's a full-grown adult. So the only way we can grow is if our cells can divide. And so we're going to talk about some of the basic concepts of that cell division. So if you just take a cell, we can divide that cell life, like a life cycle, you want to say the life cycle of a cell, the cell cycle, into two main parts, interphase and the mitotic phase, or the actual cell division phase. The interphase, that's just when the normal cell is kind of doing its thing. It's doing its normal cell functions. When you're actually dividing the cell, that's called the mitotic phase. So let's talk about what's going on when you divide a cell. Now, if you have a cell, it has DNA inside of it, and I'm going to make it double-stranded like this, okay? DNA is where we have our genes. It's the blueprint for our lives. All of our information that makes us who we are is inside of our genes, coded for in our genes. And if we wanted to divide a cell and make two, we've got to make sure we have enough DNA to fill in those two cells. So what we have to do is we have to copy the DNA first. We have to make another copy of the DNA so that each cell winds up with enough DNA. So this first step before you can even divide the cell is called DNA replication, where you basically copy the DNA. Now, in order to copy the DNA, it requires two structures. One's called DNA helicase, and one's a DNA polymerase. You may, that should be DNA helicase. Sorry about that. Helicase, DNA helicase, and DNA polymerase. This DNA helicase basically breaks or unwinds the DNA, and the polymerase builds the DNA, or makes it. So let me show you a schematic of that. Again, this is real simplified. Don't get too bogged down in the details. If you think back when we talked about organic chemistry, we talked about adenine, thymine, guanine, and cytosine being the building blocks of DNA. One thing you can notice in DNA, adenine and thymine always go together, so this yellow and green. So always, wherever there's an adenine, there'll be a thymine. And cytosine and guanine go together. So here's cytosine and guanine, cytosine and guanine. So those always are matched pairs. So what happens is, here's your DNA, and it's a double-stranded helix, which means it's wrapped around each other like a helix. And this DNA helicase comes in and unwinds the DNA, basically separates it into two separate strands. So you have strand 1 and strand 2. Now what you want to do is make a copy of the DNA. So what you take next is a DNA polymerase, and that attaches to one of the strands, and it basically matches the adenine, thymine, or the cytosine and guanine. So for example here, it's going to be moving in, this one's moving in this direction. you have a green one, which is an adenine, so you've got to bring in a thymine. Here you've got a guanine, so you've got to bring in a cytosine, and so forth and so forth and so forth, until finally you have a double-stranded DNA. And the same thing's going on the other side. Don't get bogged down in leading and lagging strand. Again, I spent an entire semester just studying cell biology, and I'm going to tell you what you need to know in 15 minutes. So just know that you have a helicase that unwinds the DNA and a DNA polymerase that then builds the DNA back, makes your copy. So your result of the DNA replication, this copying of the DNA, is you have two identical strands of DNA. Okay? If you see the words, you may come across this sometime in your reading, where it's called semi-conservative replication. That means that you don't have two completely new copies of DNA. What it means is each new copy of the DNA has one strand that's an old copy, or it's an old strand of the DNA. Let me say that again. So let me explain that. So here's your old strand, okay, and here's the new one that you're going to make, and then they're going to wind back up. So your new strands have one old strand and one new strand, so it's semi-conservative. So you don't have a brand new version of DNA. You've got some old and new that are put together to make this DNA. Alright. Now, once you have copied the DNA, then you can actually divide the cell. But there are two differences. There's two main ways that cells can divide. We call this meiosis, and let me just go and write, should be said mitosis. I've got mitotic cell division, but meiosis and mitosis. Meiosis is what we do when we make a gamete. If you don't know what a gamete is, that's basically the sperm and egg. Mitosis is what we do when we build and repair tissues and make our body bigger. So what we're going to basically focus on in this course in 201 is mitosis, how we develop and become bigger over time, and all that's due to mitotic division, where we just constantly copy our DNA and make another clone of our cell, and make another one and another one, and we get bigger. In 202, you're going to talk about meiosis and how you form sperm and egg. Now, the main difference between mitosis and meiosis is basically how much DNA you wind up with at the end. So, do you know how many chromosomes you have? All right, we all have 46 chromosomes. We have 23 pairs. So a human, a full adult human, or once you're conceived, has 46 chromosomes, or 23 pair of chromosomes. So in mitosis, you have your 46 chromosomes. You double that DNA, right? That DNA replication, so that you wind up with how many? 92. Then when you divide the cell, that gives you 2 cells with 46. And that's where you want to be. Now, sperm and egg, they don't have 46. If you had 46 chromosomes in a sperm and 46 chromosomes in the egg, and they came together, you would have 92. Well, that's too many. You can't have that. What you want is the sperm to have 23 and the egg to have 23, so that when they come together, you get 46 again. This is what meiosis does. So in mitosis, you have a cell that divides and gives you exact clone with the same amount of DNA. 46, 46, 46. Everything's exactly the same. In meiosis, you take a cell, and the end result are going to be cells that only have 23 chromosomes in them. Again, there's a lot of steps involved in that. Don't get bogged down with it now. We'll talk about it again in 202. But that's the main difference in mitosis and meiosis, is you're still dividing the cell, but you're winding up with a different amount of DNA as the end result. Again, and something else you may have heard or seen before when you talked about mitosis and meiosis were things like prophase, metaphase, anaphase, and telophase. We're not going to talk about that this semester. If you're interested, you can watch it. There's a tutorial inside Mashburn, and you can go back over that and look at what's going on in prophase, metaphase, anaphase, and telophase. I'm not going to ask you to tell me that, okay? Just know, if you see the words prophase, metaphase, anaphase, telophase, that has to do with mitosis. Another word that has to do with cell division is called cytokinesis. If you go back and think about the cell biology, you have a cell, and all that gooey stuff that's holding all those organelles together was called the what? The cytoplasm. Well, when you divide the cytoplasm, we call that cytokinesis. So in cell division, you have to divide not only the DNA, you've got to divide the stuff in the cell. So a lot of times in cell division, you have a big cell, and the daughter cells are actually going to be a little bit smaller because you've got to divide the cytoplasm. All right. So again, that's cell division in a nutshell. Don't get too bogged down with it. I just basically want you to know the difference. What does DNA replication mean? What is required for DNA to replicate? You need a helicase and a polymerase. And your end result is to basically make a clone of the parent cell, make two daughter cells that are exactly like the parent cell, so that you can grow and repair tissues. Okay? All right. If you have any questions, let me know. I'm going to move on to protein synthesis, and I'll see you again in just a second.